Whitebark pine resistance to the fungal pathogen white pine blister rust. The boreal forest is economically important in Alberta as a major industry that is responsible for $5.3 billion of revenue a year and providing 18,000 jobs. Conifer species at northern latitudes that make up the boreal forest may already be at risk in a warming climate, so it is important to do what we can to protect them from other types of harm. An economically and ecologically important group of conifer species to the boreal are the white pines. Many are considered keystone species in northern and high elevation regions because they are some of the largest and oldest trees in the world and are used for lumber as well as an ecological resource for many animals. One species in particular, whitebark pine, is of great importance. It is an early colonizer after forest fire and can tolerate cold and dry climates. It also produces large, nutritious seed cones that are eaten by many birds, squirrels, other small mammals, grizzly, and black bears. Whitebark pine is also involved in other ecological interactions. In particular, the Clark's nutcracker bird is its obligate seed disperser. This is a highly specialized relationship that is dependent on a certain abundance of whitebark pine. Nearly ubiquitously throughout the whitebark pine range is the presence of an invasive fungal pathogen, Crenartium ribicola, that causes the disease commonly known as white pine blister rust. The visible symptoms of fungal infection are cankers or sores that form on the needles and branches of the infected trees. By the time cankers are formed on needles, the tree is almost always doomed to mortality within 3 to 10 years. Whitebark pine is on the Federal Endangered Species List, and a major contributor to this designation is the infestation of white pine blister rust throughout the whitebark pine range. However, there is hope. Following tree mortality due to white pine blister rust, new trees can be planted in decimated regions that are naturally resistant to infection. Natural resistance already exists in some populations. However, because this fungal pathogen is not native to North America, there has not been as much time for coevolution, so resistance that exists is limited. Therefore, it may take some searching to find trees with natural resistance. Recent research has identified 8 genetically distinct subgroups within the whitebark pine population. It would be useful to screen these subgroups for natural resistance to the pathogen and use these genotypes in selective breeding programs to help rescue the endangered Pinus albicollis, whitebark pine, species from imposing extinction. The experiment I propose will last for 3 years. The research will be done by myself, 2 undergraduates, and a graduate student. We will obtain whitebark pine seeds from each subpopulation from the National Seed Center, and we'll have all 8 identified subpopulations with 150 replicates. 75 of these will be controls and 75 treatment. We will grow the seedlings until 8 weeks old, inoculate them with the fungus using the torn needle and paintbrush method. The control seedlings will be wounded in a similar way without fungus applied. We will watch for symptoms of infection, canker and necrosis, and biannually do a tissue harvest of needles and stem. Then, using that tissue, we will carry out qPCR fungal detection to determine if the quantity of fungus in the stem tissue and needles correlates with how many cankers are present. Counting the number of cankers to represent the degree of infection is a method that has been documented in the literature. Finally, statistical analysis will be used to determine if any subpopulation shows more resistance to the pathogen than others. The deliverables of this research would include, firstly, being able to identify genetic subgroups that are good candidates for finding genetic markers that can be used by forestry management and tree breeders. Secondly, it would be a proof of concept to determine if the number of cankers on needles correlates to how much fungal DNA is found in the tissue. And thirdly, to determine if necrosis around the canker is a sign of resistance to the fungus, as is the case in another white pine species, limber pine. Here are the references, and thank you for your attention. I now welcome discussion from the panel.